Hello everyone. So our today's topic is starvation. Let us first take an overview of fed and fasting state metabolism. So first, fed state. Fed state is 1 to 4 hours after food intake, and the dominating hormone in fed state is insulin. Insulin promotes glycolysis, inhibit gluconeogenesis, inhibit glycogenolysis, inhibit lipolysis, and also inhibit ketone body synthesis. Now the fasting state. Fasting state is further divided into 4 stages as per the time after food intake. And all these stages have different energy sources. Now the first stage is early fasting, which is 4 to 16 hours after food intake. I should write it here. So stage 1 is fasting, early fasting, which is 4 to 16 hours after food intake, and the energy source for this stage is hepatic glycogenolysis. Next stage is fasting, which occurs after 16 to 48 hours of food intake, and the energy source is gluconeogenesis. Next stage is prolonged fasting or starvation, which is 48 hours to 5 days after food intake, and the energy source is ketone body. Last stage is prolonged starvation, which occurs after 5 days of food intake, and the energy source for this stage is muscle proteolysis. So we can see that muscle proteolysis is the energy source for prolonged starvation, and that is the reason behind the loss of muscle mass in case of starvation. The dominating hormone in this state is glucagon. Action of glucagon is opposite of insulin. It inhibits glycolysis, promotes glycogenolysis, promotes gluconeogenesis, promotes lipolysis, also promotes ketone body synthesis. So the fed state is a condition in which we have enough glucose, and fasting state is a condition where we do not have enough condition, and our body then switch to various metabolic pathways in order to form glucose. So our today's topic is starvation. So we will mainly talk about starvation and ketone body synthesis. Now, hepatic glycogenolysis, as the name itself suggests, glycogenolysis means breakdown of glycogen. So, in hepatic glycogenolysis, glycogen break down into glucose in liver. It occurs after 4 to 16 hours of food intake. Hepatic term is specified here because glycogenolysis can take place at two sites, skeletal muscle and liver. But in case of starvation, liver is the major site where glycogenolysis takes place. And hepatic glycogenolysis can takes place only for 4 to 16 hours because glycogen source is limited and it cannot be used after 16 hours. So after 16 hours, our body shifts to gluconeogenesis, which occurs for 16 to 48 hours. Gluconeogenesis, as the name itself suggests, genesis means synthesis of glucose from new substrates. So these new substrates are non-carbohydrate substrates like glucogenic amino acids, lactate, glycerol, also propionyl, coenzyme A. So these are the non-carbohydrate sources, lactate, pyruvate, alanine, glycerol, and they all undergo gluconeogenesis inside liver to form glucose. So this was all about gluconeogenesis. Now comes the topic starvation. Starvation refers to the metabolic state that occurs after complete depletion of exogenous energy sources, where the body progressively shifts from carbohydrate to fat and then to protein as fuel. We have already seen here how our body shifts from carbohydrate to fat and then to protein. So now before starting ketogenesis, that is synthesis of ketone bodies, let us understand why ketone bodies are formed. As I have already told earlier that in fasting state lipolysis process takes place and this form triacylglycerol and these triacylglycerol then undergoes oxidation and form free fatty acid and these free fatty acid then again undergo beta oxidation and form acetyl coenzyme A. Normally what happens is these acetyl coenzyme A undergo TCA cycle and form energy. But in case of starvation, although we have enough acetyl coenzyme A, but it cannot get into TCA cycle because of lack of an intermediate of TCA cycle, that is oxaloacetate. This oxaloacetate undergo diversion towards gluconeogenesis in starvation, and that is why we do not have enough oxaloacetate so that acetyl coenzyme get condensed and enter into the TCA cycle. So this is the reason why acetyl coenzyme may then undergo ketogenesis and then form ketone bodies. Now let us study ketogenesis. This takes place in liver. That is, ketone bodies are formed in liver. It occurs in 5 steps. Step 1 is condensation, where two molecules of acetyl coenzyme A combine with the help of enzyme acetoacetyl coenzyme A synthase and then form acetoacetyl coenzyme A. This acetoacetyl coenzyme again combine with one molecule of acetyl coenzyme A and form HMG coenzyme A with the help of enzyme HMG coenzyme A synthase. This is an important enzyme because this is the rate limiting enzyme of ketogenesis. Now this HMG coenzyme A, which is formed, then undergo lysis with the help of enzyme HMG coenzyme A lyase and form acetoacetate and acetyl coenzyme A. And this acetoacetate is our primary ketone body. So now we finally have our ketone body, and this ketone body then undergo reduction with the help of enzyme dehydrogenase, which then form beta hydroxybutyrate, which is secondary ketone body. And then also this acetoacetate undergo spontaneous decarboxylation and form acetone, which is also secondary ketone body. We already know that acetone is a volatile substance and it gives fruity odor to breath. So this is all about ketogenesis, and we have three ketone bodies: acetoacetate, beta hydroxybutyrate, also acetone. These are secondary and acetoacetate is primary. Now comes the that now we have ketone bodies, but how these ketone bodies will ultimately provide us energy, how they will provide energy. Then there comes ketolysis, where those ketone bodies which are formed in ketogenesis undergo utilization. So acetoacetate, which is our primary ketone body, with the help of enzyme thiophorase, forms acetoacetyl coenzyme A, and this acetoacetyl coenzyme A then form acetyl coenzyme A, and this acetyl coenzyme A then undergo TCA cycle. So this is how ketone body will form energy. Now question arises that now we have energy in starvation, but who will utilize it? So we have already studied that ketone bodies are formed in liver, but they cannot be utilized by liver. I will later explain the reason. So ketone bodies are utilized by extrahepatic tissue, as I have told that liver cannot utilize it. So it will be utilized by extrahepatic tissue. First is brain. Brain derives 75% of its energy from ketone bodies during starvation. Next, kidney, heart, also skeletal muscle. Now comes the question that is there any site where ketone bodies cannot be utilized? The answer is yes, ketone bodies are not utilized at two places. First is liver because it lacks thiophorase, and second is RBC because it lacks mitochondria. Mitochondria is required for TCA cycle, and thiophorase, I have told earlier that it is required by acetoacetate to form acetoacetyl coenzyme A. So this is about ketolysis and how ketone bodies are formed and how they leads to the production of energy. Now comes the topic ketosis. So normally what happens is The rate of ketone body synthesis is enough so that extrahepatic tissue can utilize it. But when rate of ketone body synthesis exceeds, and it exceeds so much so that it cannot be utilized by extrahepatic tissue, then they get entered into the circulation, and that leads to ketonemia. And then ketonuria, also fruity breath odor because of acetone. So all these three together, that is ketonemia, ketonuria, and fruity odor breath, leads to ketosis. So now the question arises that what are the causes? First is starvation, we have already studied, and next is diabetes mellitus. We know that in diabetes, our major energy source is no, in diabetes, there is enough glucose, but it cannot be utilized because of deficiency of insulin. Therefore, lipolysis takes place. It ultimately forms acetyl coenzyme A, and these acetyl coenzyme A then undergo ketogenesis and then form ketone bodies. And when it is in excess, it then leads to ketosis. Now, the salient features of ketosis. First is metabolic acidosis because of the two product, that is acetone and beta hydroxybutyrate. Next is decreased buffer because the bicarbonate which is present get utilized in neutralizing both of these. It balance acetone and beta hydroxybutyrate. Next is Kussmaul respiration, that is acidotic breathing. Due to hyperventilation. Next is acetone breath, smell, acetone smell in breath, loss of sodium because ketone bodies are present in salt form, and dehydration. Because of loss of sodium, there will be dehydration. Now, how we will diagnose ketosis? There is one very famous test, which is Rothra's test, with the help of which we can detect presence of ketone bodies in urine. So a purple color ring is formed at this interface of ammonium hydroxide and urine. There is one differential diagnosis of ketosis, and that is diabetic ketoacidosis. And in order to differentiate it from ketosis, we have one test, that is Benedict test, which is positive due to the presence of glucose. So in case of diabetic ketoacidosis, we have both Benedict and Rothras test positive. But in case of ketosis, Benedict test is negative, as we already know that only ketone bodies is formed in ketosis, not glucose. And Rothras test is positive. So this is how we will differentiate ketosis from diabetic ketoacidosis. So this is all about starvation. Thank you.